So the next topic after studying scaling electron microscopy is the transmission electron microscopy. Well, you will find a lot of things similar here in this particular technique of spectroscopy, where we actually or in this particular characterization technique where we are visualizing the nanoscale of a material. But what happens here basically is the scanning electron microscopy takes backscattered electron or the secondary electron as the signal to be fed into the detector. But when we are talking about the transmission electron microscopy, this family of signal is different from that. So that is the basic difference between these two, because the signals which transmits through the sample is used for transmission electron microscopy, and the signal which are actually going back into the same direction from where the electrons have come, the incident electrons, will be used for studying the scanning electron microscopy. But the sample preparation and the sample should be kind of different, because what happens is the thickness of the sample doesn't play a major role when we talk about scanning electron microscopy, because it is doing a morphological characterization. But in transmission electron microscopy, the sample more or less should be thin, so that the signal can actually get transmitted. A very simple construction is something more or less similar to SEM, except from changes like where the detector is placed and so on. But the other parts more or less remains the same. For example, the electron source. So whatever electron source we have studied in case of SEM is similar in case of TEM also. So both are connected. So hence we are not going to take too many lectures to finish this particular part of the syllabus. Probably one more lecture and we should be all okay. So please listen to this carefully, okay. And if I am asking you a question on TEM, maybe regarding the paths, you can refer to SEM and give a similar kind of explanation. Then we have got the condenser lens, and the condenser lens and the objective lens, okay, and so on. Okay, these things are all nothing but the electromagnetic fields which are being applied. Then we have got the condenser aperture, okay, through which the electron actually flows through, okay, and this is can be controlled. The aperture, you can understand that basically is nothing but a very small opening. And then we have got the sample. Now once the signal or the electron falls on the sample, a number of signal gets detected, okay, the transmitted electrons and so on. Now these electrons are again allowed to pass through another lens, which is the objective lens, and then we have got the objective aperture. So then finally we are going to focus it using some more lenses, which are the intermediate lenses, and then there is something which is called the projective lens, which will basically project the signal onto the screen. So this screen may be a detector as such, okay. So this detector might be connected to a computer system which will actually form the image. So we also have something which is called the dark field imaging and the bright field imaging, which we have also seen in the previous classes. So TEM, just like SEM, is an analytical tool. It basically allows the visualization and analysis of the specimen in the realms of microspace to nanospace. So the zoom in capacity, the magnification, the resolution, these are all important parts of these electron microscopy system, okay, and they are much better than light microscopes. So a detailed microstructural examination through high resolution and high magnification imaging, we can do that, and basically it involves the investigation of crystal structures. So you have heard about crystal structures and all, okay, the lattice points, the atomic positions and so on. These things actually can be understood or studied under a very powerful microscope using the electron as a source, and as transmission electron microscopy. And then we can understand what is the specimen orientation, if there are some defects and so on, okay. And then the chemical composition of the phases, the precipitate and the contaminant. So if there is some kind of contaminant in the sample, that can also be studied using a transmission electron microscopy. And all of this is actually done by knowing the diffraction pattern. So whenever the electron beam falls on the sample, it is diffracted and it will form some kind of pattern, okay, and to follow all the laws that we actually have studied about, and then it will be nothing but give a diffraction pattern. So these things, whatever we are discussing, the investigation part, this can be studied using the diffraction pattern Of course, there will be some X-rays which are generated, which is also part of TEM, just like SEM. We can actually study the EDS pattern, and then electron energy analysis. So the electron which crosses through will actually have lesser energy than the one which has been incident, okay, for some part of it. So those electrons energy can be analyzed to know what kind of sample is the specimen. So it basically produces an image from a sample by illuminating the sample with electron within a heavy vacuum, high vacuum. So this entire setup that we are actually talking about is placed under a very high vacuum for the reasons which we have already discussed in the previous classes. We don't want additional signals and all to get produced. We don't want any ionization of air and so on. Okay. So detecting the electron through that are transmitted through the sample. So these are detecting the electron that are transmitted to the sample, and we can actually observe the column of atoms that are present in the crystalline sample. So we can actually go into the atomic scale, very much possible. Okay. In general, if you actually look into an optical image under light, okay, and look into let's say a silicon wafer. This is basically a silicon wafer. Okay. So it's basically a silicon wafer. Both the images are silicon wafer, and if you look under the optical microscope, it will look something like this. Okay, with the highest of zooms, you cannot really go into the atomic scale. But if you look it under a TEM, you will basically get these kind of images. Okay, where basically the dark region might be the region where the electron could not pass through the sample, and the light region are the regions where the sample actually the electrons are able to pass. Okay, and there is in between a gray region where the diffraction pattern can also be observed. So way the transmission electron microscopy creates a conventional image, it basically uses a bright field imaging. So whatever image we saw in the previous slide was a bright field imaging, and it can actually be compared to a show puppetry. So you basically have a puppet show where basically someone actually uses the shadow. So what happens is, let's say you have a torch. Okay, let's take an example. You have a torch and you basically shine it on a window with a lattice. So basically this might be a window. Okay, and then you might have some lattice structure there, and then you might actually shine on it. Okay, then what will happen, light will pass through the transparent part of the window. So it will pass through these transparent parts, right, these transparent parts, and not through the region where the lattice is put. Okay, and basically it will be stopped by the lattice bar. So what are you going to observe on the wall? You are going to observe the lattice bar as shown as shadows. Okay, so these lattice bars that we are seeing, this can be observed actually as shadows, where you can see there are three rows of lattice bars and 4 columns of lattice bars. Okay, so 4 columns of lattice bars. This can actually observed on the screen. Similar thing also happens in TEM. What happens is a beam of highly energetic electron instead of light. So we don't have the torchlight here, we have the electrons which will actually fall on the sample. Now some part of the sample will stop that electron, obviously, okay, or deflect the electron more than other parts. So some will deflect and some will stop. Now remember, if the sample is too thick, the atomic positions are there everywhere. Okay, so what happens is the electron might be able to go through from the first layer, some part, but from the second part it might not be able to go through. So as a result, it becomes thicker and thicker. The chances of electron transmitting to the other part becomes lesser and lesser. So after the electrons are getting transmitted, these electrons are collected and below the sample. Okay, and then we basically have a phosphorescent screen there. So we can have a direct imaging by using a phosphorescent screen, or we can actually use a camera and record it. Where basically the electron do not pass, the image is dark, quite logical, and where the electrons are unscattered, the image is brighter. So unscattered means straight away passes through without intersecting with any of the atoms. But then definitely there will be some ranges of grays in between, okay, depending on where the electron interact with and are scattered by the sample. So there may be some kind of scattering where the electron gets scattered, and it's neither too dark, neither very bright, like the region where the electron directly passes, and there is some kind of scattering or diffraction pattern which is being observed. Okay, so this is what we are talking about here. So this part where we actually talk about the backscattered electron or the emitted X-rays, or even the secondary electrons, okay, or even the secondary electrons, these are all actually used to study the SEM studies. But if you consider the TEM study, we have the electron which actually directly passes through. So these are unscattered electron beam. There is another characterization, EELS, which is used, We also have got some inelastic scattered electron which is used to TEM study, and then the elastically scattered electrons, okay, which basically loses some energy and velocity while going through the sample. That can also be used to study the SAET. So basically this is the electron diffraction pattern. So SAET is a very important study to study the electron diffraction pattern, gives a lot of information about the crystal structure and so on. The magnification using TEM can be as high as you can see it's 10 lakh times, okay, so it's 10 lakh times. So magnification, and the resolution can be as low as 1 nanometer. It's in fact below 1 nanometer, right? So we've talked about a good resolution and a bad resolution, and 1 nanometer can usually be considered as a good resolution, okay. So you can observe the difference. So if we consider an atom, they are placed very close to each other, maybe even a region less than 1 nanometer, and that actually can be understood or resolved using tunneling transmission electron microscopy. So just as SEM, it has got some advantages over the optical microscopy, most important being the resolution at high magnification. So even if you go for a very high magnification, the best possible resolution that a light microscopy can give is around just 200 nanometer, which is actually quite far if you go into the atomic scale. But if you consider TEM, the resolution is less than 1 nanometer. The structural information which is being provided, definitely light microscopy cannot provide that, okay. Even if it provides it very superficially. So if the material is being viewed as a periodic structure like a crystal, then the beam can interact with that structure in such a way that it diffracts. So basically if it has got a periodic structure, the diffraction peak will actually show some kind of pattern, and then you know basically it's a crystalline material where the atoms are placed at particular lattice points and so on, okay. So this basically provides a lot of information on the crystal structure, the symmetry and the orientation of the material. Moreover, the micro analysis can be done. That means in fact we can also talk about something which is called the chemical composition using TEM. So please remember, TEM usually doesn't give you any kind of colored images. This is basically nothing but a contrast of different electron images, okay, the signals. So the parts of the machine, we are not going to discuss these parts, okay, because we have already seen it in SEM, okay, and these more details actually can be looked into when you actually go and really use these kind of instruments, okay. But I think the basics are already clear to you. So electron gun is there at the top, which is similar to SEM, and the entire electron column, okay, is basically placed under the vacuum for obvious reasons which we have already studied. And then there is the electromagnetic lens system similar to SEM. So we have got the different lenses which we have seen, okay. So these are actually nothing but used for collimating and making the signal better and focusing and so on. And then we have got the detector. So we can have a scintillation screen, or we can have a camera and so on. And then we have got the water chilling system. Now this basically heats up because we know that we are using magnetic lenses and we are using some kind of electric field, and as a result the heating up of the system is you cannot avoid that. So you need to keep the system cold, so use a water chilling system. Most of the times you will also see that these kind of setups like SEM, TEM is actually kept in a room which has actually got a AC provision, keeping the weather conditions in mind. The specimen and the sample chambers are there where the specimen is placed before it can be studied using the electron gun, and then the main control panel and the operational controls, and of course the image capture. So these are the important parts, okay, of TEM instrument. And if you look into a real TEM instrument, there are lots of controls there, okay. So you basically can adjust, okay. So this is basically nothing but works like a mouse. You can adjust the different positions, and then there are different setups, okay. So if you look into a typical TEM setup, you will go into these kind of controls that you should actually know. So a professional who works on a TEM instrument will be knowing all about these, okay. We're not going to go into the detail of this. This is a setup of a Geol company, okay, one of the prominent companies which produces a TEM machine. But then it has also got some drawbacks which we cannot avoid, okay. TEM cannot take colored images. So basically, as I told you, it basically has only the images which are black and white. So if you want to have a colored image, they can be actually added artificially using some softwares, okay. So for example, you have got a system where you have got more atoms, so you can add a different color to it. You have got less atom in the same system, you can add less color to it. So in that way you can differentiate with where the density is more, where the density is less, and so on. These kind of studies. TEM again cannot image through the thick sample. The very first slide I told you, usually the sample thickness is around 100 to 200 nanometer. Thicker than that, the electrons usually cannot penetrate, okay, and then we can actually cannot have any kind of signal to do the TEM study. A standard TEM cannot image surface information, so it basically doesn't give any kind of surface information. Surface morphology is more or less studied using SEM. A TEM cannot reliably image charged molecules. Now basically these are electrons which are coming in, and these are charged particles. So if there are any kind of charged molecules within the system, let's say an ion, let's say a species of Na plus exists, okay, it basically becomes volatile under the electron beam, because the electron beam will basically exert a force on the charged material. So if it is Na plus, electron beams are negative, and then basically it will exert a force, and as a result the signal that you are going to get is not going to be enhanced, or it's going to get lost, or it's going to get scattered too much, and so on, okay. So what we need is we don't want a system which is actually having charged molecules in it. That cannot be studied using this, okay. So that's all for today's class on TEM. We are going to see into a bit of detail on how the sample is prepared for TEM analysis in the next lecture. Thank you.